السلام عليكم ورحمة الله وبركاته. Can you hear me?
Alaikum assalam. Yes, we can hear you.
Okay. Bismillah ar-Rahman ar-Rahim. Today, inshallah, we are starting paper 6. How to solve paper 6. I'm giving you some tips. Inshallah, starting from tomorrow, Friday, also, I will start giving classes on how to solve paper 6. Okay? These are some tips and tricks to solve paper 6 and everything related to it, inshallah. Okay? طيب. What about paper 6 and how to solve paper 6 and everything related to paper 6? Okay? Let's start. Bismillah ar-Rahman ar-Rahim. Paper 6 is basically formed from 40 marks. It has 40 marks. And you are going to solve it in 45 in 1 hour. I'm sorry. Okay? And take care. Paper 2, paper 4, paper 6. Each one has a factor to be multiplied with. Except paper 6. The mark that you get in paper 6 is the one that comes out in the total of 200. So paper 6 is very important. Mainly paper 6 comes in two questions, question 1 and question 2. Mainly question 1 comes as experiment, drawing table, graph analysis. These are the main questions that comes in question 1. Question 2 mainly comes as investigation question, and we will learn how to do it. It also comes as drawing a graph, draw a graph, and comparison, how to compare between two organisms or between two species. Okay, so this is mainly the main idea of paper 6. Before we go into paper 6, you have to understand some command words and how to understand them. We will go into two parts, explain and sketch and scribe. When he asks you to explain, this means that you are giving a reason for the results that have been given to you. When he asks you to describe, you just use the diagrams, use the graph, use whatever he has given to you, use the figure that he has given to you to give your statement. Sketch is the one which is in paper 6, which is drawing. And we will start, inshallah, covering it now. This start with the first one, which is very important, graphs. How to draw graphs. First of all, you have to know that graphs are three types. Could be a straight line, could be a linear curve, one, or it could be broken line. Mainly, curve and broken line comes in the exam. But whenever I draw a graph, I have x-axis and I have y-axis. Who is always on the x-axis? To understand this better, you have to know what's the difference between dependent factor and independent factor. Who can type for me in the chat? What's the difference between dependent and independent factor? Yeah, who can tell me? I'm waiting for your answers. Who can tell me in the chat the difference between dependent and independent? Can you hear me? Hello? Do you hear my voice?
Yes, yes.
Okay, who can tell me the difference? Or you can tell me verbal, no problem. Difference between dependent and independent factors. If you know, just raise your hand. Yes, yeah.
Independent factor is what you are testing for. For example, when uh you're testing for
Temperature, for example.
yeah, different temperatures. Uh you're testing the enzyme activity of the different temperatures. The different temperatures is an independent variable, and
Yes,
then enzyme activity is a dependent variable, which is relying on the temperature.
excellent. So independent factor is always on the x-axis, while dependent factor is always on the y-axis whenever you draw a graph. Also, when it comes in a table, the one which is on the left side always on the x-axis. This is a tip that you have to know. And the y-axis on the right side is always the dependent factor. Very important to know. Some questions comes as a line of best fit, but very rare to come. A line of best fit you have been through before. It's a line does not pass through all the points, but it shows the general trend. At least it passes through the majority of the points. Doesn't come also as a graph all the time. Sometimes it comes as bar chart or histogram. This is very important. Very important to understand the difference between bar chart and histograms. First of all, in bar chart, bars are not touching each other. Okay, bars are not touching the y-axis. Bars should be evenly spaced between each other. Yeah, مثلا for example, you give 1 centimeter for bar chart, 1 centimeter for space. 1 centimeter bar chart, 1 centimeter for space. 1 centimeter one chart, 1 centimeter for space. And we will practice this inshallah. But you have to understand the rule, which is what? Not touching each other, not touching the y-axis. Spaces and the width are same. Next, in a histogram, this is very important. They are touching each other, no problem. And they must be the same width, which means what? Don't draw the first histogram with 1 centimeter and then you do the second with 2 centimeter. This is wrong. Okay, and they are not touching the y-axis. So till now in graphs, you have learned that x independent variable, y dependent variable, bar chart evenly spaces, not touching the y-axis, histogram touching the y-axis, even width and no spaces. Okay. Okay. Next, how to plot a graph. How to plot a graph. Independent on the x-axis, dependent on the y-axis. When you plot a point, okay, مثلا for example, this is the x-axis, this is the y-axis. I need to plot this point. I don't do only a point. This is wrong. You do an X or a dot surrounding with it a circle. Very important. Number two, you have to write the head. What is this? What are you talking about this? Who is written here? Is it temperature or whatever it is? Another point that you lose marks in, you have to write the unit of the factor that you're talking about. Here also the same. You have to write the factor and also you have to write the unit. Very important. Another rule, you have to use a scale. What does the scale mean? Should be equivalent scale, and we'll practice how to make a good scale, inshallah. Point number three: your graph must pass more than 50% of the space given. Yeah, مثلا for example, don't draw your graph here. This is wrong. The graph should extend more than 50%. So you should use your scale wisely. Next, you have to label the axis with units. Very important point after you finish this part, you have to mention a key. When do you mention a key? If there are more than one point is repeated, or there are more than one factor needed. Now, مثلا for example, sometimes, مثلا for example, I'm sorry. Sometimes is asking for what? Is asking you to plot, plot what? Plot a temperature, plot pressure, for example, plot transpiration. Any three things needed to be mentioned. So you have to mention a key. A key which means what? That this one is a temperature. The one which is broken line, this is for example pressure, whatever. The one which with X, it's the light intensity. It's called a key. And we will, inshallah, will talk about it when we solve the past bit. Just I need you to be sure that you are giving the basics. Today we are giving the basics. Next, whenever you describe a graph, so I have prepared this for you. In dark landscape, smart printers find treasure, which means what? I stands for independent variable on X, dark dependent variable on Y, L you label with units, S must use appropriate scale, P plot the points as X or O, F fit the best line or the curve or whatever. You should join the lines with ruler. Please take a screenshot for this. Take a screenshot for how to plot a graph. This key will help you a lot. Take a screenshot. Done? If you have taken a screenshot, please raise
No,
your
wait,
hand.
sir. Wait 1 second.
Take a screenshot on how to solve or how to plot a graph. Okay. Next question comes in graph is how to describe a graph. To describe a graph, you have mentioned 3 points. Number one, you have to mention the trend. And مثلا for example, as X increase, as temperature increase, what happens to the Y? Is this one increasing and this one increasing? Is this one decreasing, this one increasing? This is called a trend. Number two, you have to mention the peak. Where is the peak? And you have to understand how to get it. You have to mention the lowest point for enzyme activity, for example. When it's declining, when it's increasing, the steep slope, the steep decrease, is it gradual increasing? Is it gradual decreasing? Yeah, مثلا for example, here we have a graph. This is called, look, a steep increase, decrease, I'm sorry. This is a steep increase, this is a steep decrease. طيب for another graph, let's compare between gradual increase and gradual. This is called gradual increase. This is called gradual decrease. You have to mention the most quotation point in your graph. Next, you have to mention when it's constant. So to understand how to describe a graph, I memorized you in this. This is what? This is a key on how to, just a minute. Yes, like this. This is what? How to answer a question. No, thank you. I will now reopen again because, yes, how to describe a graph. You use this technique, SQLPT. Whenever he asks you to describe a question or a graph, you mention this. SQLPT. S, the slope. Is it steep, gradual, curved, flat, increasing, decreasing? Is it steady? Is it constant? Number two, you mentioned the quotation. The highest point was at 10 centimeter, the lowest point was, and so on. You mentioned quotation. Number three, you mentioned the peaks, the minimum. The plateau started to be constant from this point to this point. Okay. Pattern. What does it mean? The steep slope, the and so on. The trend as this increase, this decrease. Please take a screenshot for this. This is how to describe any graph. Mention these points and it will help you a lot. This also can be for paper 4. Thank you. This also can be used for paper 4. The same technique. Okay, here is a question on how to plot a graph. First of all, I will move fast. This is on the left side, X or Y axis? X, this one, Y axis. So you draw your, and so on. You write here what? the percentage concentration of solution. Is I have given you a unit? No unit is given, but here there is a unit is given. So this one, you write here the title and you write here the unit. Then you start your scale. Then you start plotting X or sample, like a point with circle. And then you ask the to describe. And sometimes, a lot of times, he asks you to get, مثلا, for example, at number 4, what was the rate of transpiration? You go up, then you mention a point. We will solve this, inshallah. Next, something called tally count. Tally count is also important. What's tally? A tally is a simple way for recording and counting number. Yeah, مثلا, for example, here we have a text. This text has some words as walk, bike, bus, walk, and so on. He wants you to answer how many times walk word was repeated. So here walk is there. So you make a line like this. Walk also there. You make a line like this. Walk also there. You make a line like this. Walk also there. So 4 times till now. Walk also there. So 5 times. You get the idea? You count it by 5 tally counts. It's called like this. Yeah, another walk. So walk here. Another walk. Walk here. Another walk. Walk here. Another walk. Walk here. Another walk. So one, two, three, four, five. You keep doing we can say the set. A set is from 5. So walk was mentioned how many times? This is one set equals to 5. Second set equals to 5 and 3. So 5, 5, 3. So the frequency of walk word was repeated in 13 times. Up here, let's go for bus. Bus was mentioned 5 times and 2. So 5 plus 2 equals to 7. Car, 4. Bike, 5. Train, 1. Do you get it? طيب. Some students investigated the variation in the number of spines of holy leaves. The figure shows 20 holy leaves that they collected from the same tree. Take care. In tally count, uh, comes, for example, in 3 or 4 marks. There is variety because the counting of one student is different than the other. But there is a range that you move through. For example, number of spines. The leaves which have 6 or fewer. Yeah, مثلا, for example, this leaf. How many spines are there? 1, 2, 3, 4, 5, 6. So we have 1. This leaf. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. 11, which is this one. And so on. You keep counting the number of leaves. 1, 2, 3, 4, 5, 6, 7, 8. No, this one's not count. 8, 9, 10. 10. So you count this one. You get the idea? And you keep counting them and you give him the total number of leaves. Yeah, مثلا, if you count it like this, so how many leaves? 4 leaves are there. Okay? This is 4 tally count. It comes with simple questions, don't worry. Next, how to draw a table. After the first question, which is the experiment one, comes something called draw a table for the result which has been given. He gives you results and then you ask to draw a table. مثلا, for example, he's telling you a student was doing an experiment and the solution was observed with different colors. مثلا, for example, the first test tube was called P. It has purple color. The second test tube was called Q. It has also purple color. The third test tube was called blue. It has R was called R with blue color. So there were 3 test tube P, test tube Q, which test tube R. The color here was purple. The color here was purple. The color here was blue. So he asks you to do what? Draw a table to show the result. So whenever you go for these questions for drawing a table, you have to mention or have to know these points. Number one, you draw the table using a ruler. You don't use your hand. Zero. Number two, you put the independent variable on the x-axis, which is on the x-axis on the left side of the table. And you write a table. Okay. Whenever you write the table, you put the independent variable on the left axis, on the left side. You put the dependent on the right side. You put headings. Yeah, مثلا for example, these are what? Colors, right? Which is was observed. So you mention what? Observation. All color noticed. In this case, in the heading part, it's more AVP. يعني any valid point from the student perspective, but of course it has a range. Yeah, don't tell me it's a color for fruit. No, it's color for a reaction that has been through. So whenever we go for colors, you say observation. Whenever you go for يعني مثلا centimeter, you say length. Whenever you go for percentage or you go for rate. Do you get the idea? You have to, when you solve exams, you will be able to understand how to mention headings properly. Next, you mention also the unit in the heading. Yeah, مثلا for example, in a heading you said it's the length for uh, for anything, for uh, for the potato, how much it was increased after osmosis. You put it in millimeter or centimeter according to the given one. Next, very important point, most of the students, they lose marks in it. You give it to the decimal place. What does it mean? مثلا for example, he has tell you that in the first experiment, the rate was 0.01 second. The second experiment, the rate was 0.02 seconds. And the third experiment, you will observe. You will count seconds or minutes or whatever, or minutes. You have to mention to the same decimal place. Okay? Sometimes he mentions zero point 0.0. So when you mention, you should mention 1.0, 2.0. You have to keep the same decimal place. I need just your confirmation for this point. Is this point, you understand? Because this point is very important. Most of the students, if you understand it, please raise your hand. Most of the students lose marks in this, that they don't give the number to the nearest decimal place. Okay? Next. So let's go for this table together. Draw a table. This is a question with column heading to show how you would display your readings. You are not required to enter any reading in the table. Explain how would use the reading to reach a conclusion. What is the reading here? There is a hot metal container. This is hot water, and this is a thermometer. A thermometer will measure what? Temperature. Type. What are we measuring here? What's my independent variable? What I'm waiting for? Yes, time. So the time will be on the left side. I write time as a heading and I give it a unit. Okay. Next, what I'm going to measure, يعني every 1 minute, every 2 minutes, every 3 minutes, every 4 minutes, what I'm going to measure? The temperature, which is my dependent variable. So I mention also the unit, Celsius. Then I take my readings according to the experiment. Also, I'm telling you this as basics. We will solve a lot of past papers, inshallah, where you are going to apply this. Type. To understand a table, very important, please take a screenshot. This is a key. Really, X-ray yield helpful, useful data. Really, use a ruler. X-ray, X independent, yield Y dependent on the right. Very important. Helpful, add headings. Useful, add units. Data, give decimal places. Take a screenshot. This is how to draw a table. This is how to draw a table. Very important. Next, some calculations also are needed in the exam. First one is easy. You already know how to get the image size of the specimen, how to convert between units, centimeter to milli, milli to micro. Nano is not mentioned. How to get the mean. You get the sum of all values over the number of all values. Very important. The mean also is mentioned. The rate. There is a difference between rate and percentage change. Okay. Type. A rate of the reaction equals to the slope of the curve, change in the axis over change in the y-axis, يعني difference in y over x-axis. Okay? Next, how to get percentage change. New value minus old value over old value multiplied by 100. A decrease, percentage decrease, old minus new over old by multiplied by 100. Percentage change final minus original over original multiplied by 100. Percentage, the decimal places multiplied by 100. Also, we will solve this in the past paper. How to get the area and surface area? Not that much mentioned, but you have to keep in mind how to get the volume. Also, you have to get in mind. Please take screenshot for calculations. If you take screenshot, raise your hand so I can pass the slide. Okay. Calculations also are very important. Okay. Also for the paper 6, you have to mention diffusion. You have to understand osmosis and the osmosis experiment, and you have to know why the plant gains mass, why the plant loses mass, why there is no overall change, when the cells are turgid, when the cells are flaccid, when the cells are plasmolized. I will share with you the file. Also in animal tissue, you have to mention when the cell is shriveled or crinated. You have to mention when the cell explode and swell. You have to mention when the cell stays the same with no change. You have to investigate the dialysis tube. Very important. يعني مثلا for example, here we have water and here we have the dialysis tube and here we have concentrated sugar solution. So the water is what? High water potential. The concentrated solution is what? Low water potential. So osmosis is the movement of water from high water potential to low water potential through partially permeable membrane. So the water will diffuse into the dialysis tube. So the tube becomes increase in size and the water level increase. In these cases, he gives you a reading for the water level initial, then a reading for the final water level, and then he asks for the rate, how much percentage change, and so on. You get the idea? Will explain and we will go through a lot of paper 6 questions related to this. Also, the enzyme activity, very important. I just am giving you some important points. Next, very important to know food tests. Food test type. Please go for food test, please. Number one, test for glucose or reducing sugar using Benedict's reagent. Very important to understand the steps. It always comes in the exam. Mention the reagent or mention the result or mention the method. If he asks for the reagent, you just say Benedict. If he asks for the result, positive or negative, you mention what color it changed. If he asks for the method, you have to mention these steps. We have been through it before, but inshallah that I share with you. Next, test for starch. Add a sample of a clean test and dry test tube. Then use iodine solution and always try to mention what mention quantities. يعني مثلا add 2 milli, add 1 milli, and so on. Add drops or 3 drops of iodine solution. A positive test always gives you color, a negative test gives you another color. Test for protein. I will share with you the tests, and of course, you know them. We're going for the most important parts. Test for emulsion, very important. Test for vitamin C, DCP/IP, most of you forget it. Add 2 centimeter cube of the sample needed to be tested. Add 1 centimeter cube of DCP/IP. Positive test will change the blue color into colorless. A negative result gives the test tube stays the same. Next, this table is very important. Take a screenshot. This table summarizes elements, monomers, polymer, food test, positive and negative result. Take a screenshot and raise your hand that you have taken so I can pass the slide. Type. For carbohydrate, What do we use? We use starch or for simple sugar or reducing sugar. For starch, we use iodine test. For a positive result, gives me blue black. For a negative result, gives me orange brown color. For simple sugar, we use Benedict, which in positive result gives me orange or brick red. In a negative result, gives me blue. For proteins, use burette. Very important point in Benedict. I was forget to tell you. Heat is needed. Please don't forget this point that you must give heat for Benedict. Heat using a water bath. Okay. For burette, no heat is needed. For a positive result gives me from violet or purple. Negative result gives me blue. For fats, we use in ethanol emulsion test. The positive result gives me cloudy or milky emulsion. Negative stays the same. Okay, can you hear me?
Yes, we can still hear you. Your
Yes. Yeah.
screen stopped sharing.
Okay. Right. Okay. This is very important. For vitamin C, we have been through this. Right. Drawing. Very important question comes in the exam, which is to draw. It gives you any space. There is not one which is mentioned or needed, and you have to understand and you have to memorize. No. Last session, strawberry came. It was first time to come, and most of the students were confused about it. But when you mention and you follow these rules, I assure you that you get the 4 marks. Take care from this. Very important trick. Drawing questions comes in 4 marks. Sometimes it comes in 5 marks. But when it comes to 5 marks, you have to make label because some students they miss to label. Do you do a very good drawing, but they miss to label. Type number one, use a pencil, mechanical one or HB. Don't ever shade. Don't make double lines. Okay, the drawing should be large to fit your space provided. I will give you a trick. For example, he gives you a cell. You measure the cell. Let's say it's 5 centimeter. Then he gives you a space here to draw. Can you see this 5 centimeter? You measure with the ruler the width and the length. Let's say that the width is 5 centimeter and the length is, let's say, 7. When you start drawing it, just add 2. Add 2. It will be better. So you do the width 7 and you do the length what? 9 or 10. Why? Because in the marking scheme, he always mention the width. يعني مثلا for example, he says not less than 6 centimeter, for example. Do you get it? طيب. When you label, is there is a nucleus, don't say and make an arrow like this. Wrong. You label with a line, not with an arrow head. The ratio should be the same. What's meant by the ratio? Whenever we have a cell like this, okay, I will tell you now. There is an apple, and you draw ratios. What's meant by ratios? Yani you split the drawing into 4 quarters. 1, 2, 3, 4. When you draw your drawing and you do the same ratios, you have to mention the same parts. Yeah, مثلا there was a seed here, 2 seeds here, 4 seeds here, and 1 seed here. You have to do the same. When you do your drawing, don't draw 4 seeds here in the first quarter. Habibi, it already was 1. Why you're mentioning 4? So your ratios are not correct. So here in quarter 3, there were 2 seeds. In quarter 4, there were 4 seeds. Quarter 2, there was 1 seed. Do you get the idea? Very important this point. The ratio should be the same. Okay? Don't forget to label. Don't write in your drawing. And if he mentioned to, مثلا for example, write a specific detail, you have to mention it. So, okay. So to better understand how to draw, I prepared for you this key. Take a screenshot. Penguins do big straight labeled diagrams exactly. Penguin use pencil. Don't use a pen. Do definite outline, no shading. We will practice this. Big, it should be big as fits the space. Straight label lines, no arrows. Labeled, add at least one or two label if mentioned. Diagram, make it double the original size. Exactly count every structure precisely, which means what? يعني the number of seats should be the same, number of layers should be the same, number of leaves in a flower should be the same. Don't, if the original is three, don't do I'm sorry, number of petals and number of leaves in a leaflet. Okay, we'll practice this. Next, compare between two figures. Just if you understand, please raise your hand. Everything is clear? Okay. That's these key points are the extract of the previous exams. If you follow these points, you will be able to solve any past paper in paper six. And trust me, paper six is the only paper that you can get the full mark easily, and you don't give yourself that much time to compare between two figures, which means what most of the exam questions he comes to ask you about to compare between two figures. Emerson, for example, he gives you two leaves, a leaf like this and a leaf like this, and he asks you to uh, when he asks you when he asks you to compare between two figures, you go for this method. What does this method say? SSNN cell wall. SNSSNN cell wall. The first thing that you look at to compare is the size. This is small, this is large. The shape. This is oval, this is palmate. The color. It's not mentioned here, but maybe it's mentioned. The number. More venation, less venation. Sometimes you have a leaflet. Do you know what a leaflet means? It's a leaf formed from smaller leaves. We call it leaflets. يعني هو here formed from leaflets, here formed from one leaf. The wall of a fruit or a vessel, thick or thin. Okay. C stands for color. L stands for here the second L, number of layers, number of leaves. L stands for the lumen. Very sometimes it comes the lumen. What's meant by the lumen? The cavity inside. Here it has narrow lumen, and here it has wide lumen. Do you get the idea? Very important to understand how to compare, and it comes in most of the questions. Type. Last part I will talk about today is how to plan an investigation. These questions most of the students get confused in, and it's a question of 6 marks. And I dare that if you understand the one that I'm going to tell you now, I dare you that you don't get 6 marks in this question. I dare. Very important technique. Please take a screenshot before we go into. I will tell you right now a very important technique. Easy technique. Very easy technique that you, when you follow, you will start to solve any investigation question in a blink of an eye. Trust me. You just remember this. Turtle fight snakes. A turtle always fights with a snake for many incredible super rewards. If you memorize this one, you got it. Turtles fight snakes for many incredible super rewards. It is the easiest question and the question that you get all If you even don't understand what does the investigation is about, and you mention some points here, I'm going to tell you, you will get the at least 4 to 5 marks. Look, number one, turtle, which means what? Starts for trial. Whenever he's telling you, يعني مثلا for example, investigate the effect of temperature on the action of enzyme. The first step that you say, please repeat, make more than one replicate. The first thing that you say, okay, the first step that you write, prepare 3 test tube at least. You got 1 point. Prepare 3 test tube. This is called trials or more than one trial because you are going to investigate the effect of temperature, right? So the first trial or the first test tube, it will be, for example, 20 Celsius, 30 Celsius, 40 Celsius. So you got the first point. You repeat the experiment multiple times at different temperature, 10, 20, 30, to ensure consistent result, reliable result. This is the first step. If you understand the first step, please raise your hand. So the first thing, the first sentence that you write in any investigation question, write replicates, write trials. Type. If it asks you, investigate the effect of osmosis on potato, you'll prepare 3 test tubes. Cut equal sizes of potato. Add in test tube 1, test tube 3, test tube 2, and so on. So the first one you always go for is trials. Number two, which is turtle fight. The first factor that you will write, the factor that you change it. Which one are you going to change here? The temperature or the action of the enzyme? You have first to understand for the action of the enzyme. Where is it? You get the idea? So here, the temperature is affecting the action. So the temperature is my independent, the action is my dependent. So you will write this. The independent variable in this experiment is the one which is temperature. The factor, don't say change it, say the dependent, independent factor. So you get the second point. يعني your ability to understand what is the independent, you got a point. Go for the second F, fight snake 4. Second, you will mention the dependent variable. Your ability to understand what is the dependent variable here, you got a third point. The dependent variable which was measured is the rate of enzyme activity. Type. Next, a very important point called S. Same, same. Same, same. What does it mean? Same, same. What are you going to keep the same? If you are going to investigate the temperature on the enzyme, what else should be the same? What else should be the same? Should I change more than one factor? No, you change only one factor. So pH should be the same. Substrate should be the same. Time should be the same. So number one, you mentioned the trial. Number two, you mentioned the factor. Number three, you mention what will be controlled, and you don't say same factor. You say controlled factor to get the point. Then you mention the dependent variable. Then you mention the method, which is many. Method very important. A method. What's method? To get a mark in the method, which means what? How you are going to measure the dependent variable. يعني مثلا for example, here I'm going to investigate the effect of peroxide. I'm going to investigate the effect of cell respiration. So I have to know what I'm going to measure and how I'm going to measure. And we will talk about it inshallah. How to measure the dependent factor, whether it is rate, whether it is temperature, whether it is oxygen released, whether it is carbon dioxide released. I have to know how to mention, how to measure it. طب let's go for this example. This method, you should add the enzyme in a solution and the substrate and measure the rate of the reaction over 5 minutes using a calorimeter, for example. For if we're going for enzyme and this enzyme is producing gas, Because it's breaking something, you will measure the volume of gas produced and so on. We will have a lot of practice. I will give you another example. Next, incredible. Incredible means what? This one here. Incredible. Improvement. How to improve this? I will give you a list on improvement in a while. Safety. How to give safety precaution? I will give you a list for safety. You will put improvement and safety in anything, in any experiment. So all of these you are getting marks. A point which is you add without thinking and you get a mark. Repeat the experiment 3 times and get and record your result and get a mean for result. Very easy. Type. Let's have a real question. This question is telling you what? Investigate the effect of light intensity on the rate of photosynthesis of aquatic plant. Before I start, what do I say? Turtles fight snakes for many credible super rewards. T trials. Please, in your experiment, repeat the give experiment and reliable result. Prepare 3 test tubes and add the same aquatic plant with different distance from a lamp. 10 centimeter, 20 centimeter, 30 centimeter from the lamp. So you are changing what? The light intensity. So this is my independent variable. You're getting to know what the rate of photosynthesis, which is dependent factor. How to know the rate of photosynthesis? By, yes, think by oxygen amount or volume of oxygen gas produced. So what's the factor that I change? The factor changed is the light intensity. You got a mark. How we are going to change it? By changing the distance. Next, who will be controlled? You're now investigating light intensity. Are you going to control temperature? Yes. Water? Yes. Same plant? Yes. pH? Yes. Everything else will be mentioned. Factors to be measured. What are you going to measure? You're going to measure the dependent variable, which is the rate of photosynthesis, and we are going to count the number of oxygen bubbles per minute. Or we are going to investigate the volume of oxygen. We are going to measure the volume of oxygen produced using gas syringe. Next, method. The method, which means what? How you are going to measure the dependent variable. Place the aquatic plant in a beaker, give it different light intensities by distance, measure the oxygen produced over time, every 1 minute, 2 minutes, 3 minutes, 4 minutes. How to improve this? Yeah, مثلا for example, counting number of bubbles is not something accurate. You should use gas syringe, and we will explain how to improve things. Safety. Anything you add, wear gloves, goggles, lab coat, take care from stains, light, heat of the lamp, a sentence that you add and you get a mark. You repeat the experiment and take your reading, get your mean. That's it. So whenever comes an investigation question, you mention what? Turtles fight snakes for many incredible super rewards, and you will get the mark that you need. Okay? طيب, Mister, for the improvement part, safety part, how to know? These are some source of error and these are some improvement. Very important to know. For example, we mentioned that we are going to number of bubbles count. This is a source of error. How to improve it? How to improve it? Use a gas syringe, use a measuring cylinder. Do you get the idea? Take a screenshot for this. These are what? These are safety precautions. Wear lab coat, for example. While cutting a knife, cut away from your finger. You get the idea? Take a screenshot, please. Some hazards, which are safety control. When you cut away from your hand. Hydrogen peroxide screws, wear gloves and goggles. Okay, take care from staining of your clothes. So all of these, you are adding it in which one? In the safety precaution part. Type this table, you add it where? Here, improvement part. You have to understand and memorize these ones. Type. If I'm going for gas syringe, why? To measure the volume of, I'm sorry, gas syringe or delivery tube, measure the volume of gases needed or liquids. Ruler, to measure the foam produced. Of course, if you have been through some past paper, there is foam produced. Next, these are some methods and their controls and how to use them. You have to mention this. Yeah, مثلا for example, how to control temperature. Very common question. Using thermostatically regulated water bath. How to control the pH using buffer solution. How to control the volume of solution measuring cylinder or gas seepage. Okay. Next, خلاص, we have been through these before. How to change the temperature using thermostatically thermostat. How to change humidity using plastic bag. How to change light intensity. Give the lamp on different distance. Airflow using a fan. Carbon dioxide concentration using what? Sodium hydrogen carbonate solution. Take a screenshot from this also. Easy. These are most of the steps or most of the things needed for you to understand the paper 6 question. Source of error comes a lot in the first question, which is experiment question, called source of error. Type. Let's have any paper here, for example. Think I opened this one. We don't need mark scheme, we need question. Any paper question. Let's go for June 2025, paper 6. The question, the paper 6. Yeah. Here, let's go for investigation question. This one. Plan an investigation to determine the effect of temperature on osmosis in potato plant. Wait, I will first download it. طيب. Remember with me the rule. Open the rule, please. The rule that we said. Turtles fight snakes for many incredible super rewards. The rule. Give me 1 minute. I'm opening it. Open with me the paper. Okay. Not opening. Are you with me? Okay. Okay, let's share the screen. Okay. Now it's podcast. So, where is the question? Here you can find the question. Bismillah. Plan an investigation. to determine the effect of temperature on osmosis. يلا. Turtles fight snakes for many super incredible rewards. What is the first step that we can say? Huh? Replicates. Prepare. Let me check here. Is it opening? It should it opens only one. طيب. This. يلا. Okay. So prepare. Three test tubes for investigating effect of temperature. First test tube with 20 Celsius, 30 Celsius, 40 Celsius, for example. Who's my independent temperature? Who's my dependent? Huh? The rate of osmosis, which will be what? The size. Okay. Next, the independent factor in this investigation is temperature, as it will be controlled by setting three different temperature we have mentioned before: 20, 30, 40. Next, S, same, control the variable. What could be controlled here? What can we control? Ah, pH, right? What else? Size of potato. What else? Mention anything that can be controlled. So here, he mentioned what can be controlled. Variable kept constant. Can you see? Type, species, age of potato, plant tissue, area, mass, surface, size, concentration of the solution, the water potential, the time left for 1 hour. He gives you 2 points only for constant. Okay, let's continue. The second factor, what is the factor? The dependent factor in this experiment is what? What he was mentioned. What will be the experiment, the dependent factor? What do you think? As we mentioned before, the mass of potato, will it change? The volume of potato, will it change? The length of potato, will it change? So the dependent factor in this experiment is the change in mass or volume of potato. Next, M. M stands for what? The method. How are you going to make the experiment? How you are going to measure this? Okay? How are you going to do this? By giving these things. You will mention what? Cut the potato in equal sizes or equal lengths. You have to mention the time. You're going to leave it in a solution for how much time. Then add each piece of potato cut, which are the same in the different three test tubes for 1 hour, for 30 minutes, whatever you are going to say. Then how you are going to say the size, how you are going to measure the size. Balance, using a balance. Measure before and after. Calculate the change in mass, or if you're going to say length, measure with a ruler before and after. You get the idea? I'm explaining for you how to use the method. Let's get back to the marking scheme. The marking scheme says what? Drying the water before weighing. Then use a potato or piece of potato. Peel the potato, cut the potato in pieces, add them in the solution. Make sure the potato is covered by the solution or submerged. Method of maintaining at least one of the temperature. Very important. How you are going to maintain the temperature. Can you remember thermostatically? Water
Insulation.
bath. Water bath. Thermostatically, water bath is used to maintain the temperature. You said that you're going to keep it in 20 and 30 and 40. How? Using this thermostat. Next, how we are going to mention the safety. Yeah, wear goggles, wear gloves. When you cut with knife, take care to cut your finger or cut away from your finger. Improvement. You have to use thermostatically thermostat, water bath to control the temperature. Or repeat, take your result and give a mean for your result. Repeat 3 times and give a mean. طب let's check the mark scheme. Variable kept constant خلاص. At least 3 trials. Repeat investigation at least twice. Relevant safety measures. Okay. This is how to answer the investigation question, and we will go through more, inshallah, these investigation question. Now I will open